Hello everyone, I'm David Hills from Separation Science, and I'm delighted to be able to welcome you to our latest expert discussion event titled Glycan Analysis in Biopharma, Challenges and Future Directions, in association with Agilent Technologies. Now do note that you can submit questions at any point during the presentation by clicking the Ask a Question button below the player window, typing in your questions and clicking Submit. All submitted questions will be forwarded to our experts after the event today, and your submitted questions will not be seen by anyone else, so don't be shy about asking them. So before we begin today's event, let me briefly introduce our expert panelists. You'll first hear from Arnaud Delobel, R&D and Innovation Director at Quality Assistance. Arnaud graduated from ECPM Strasbourg and specialised in mass spectrometry of proteins and peptides during his PhD at the CNRS in Gif-sur-Yvette in France. After a postdoc in Liège, Belgium, which focused on proteomics, he joined Quality Assistance in 2006 and held several positions in the company to eventually become R&D and Innovations Director in 2016. As the head of the R&D department, Arnaud is responsible for supervising internal R&D projects aimed at implementing new analytical technologies that will ultimately be provided by Quality Assistance to their customers. Joining Arnaud is Gordon Lausch. Gordon is the Professor of Biochemistry and Molecular Biology at the University of Zagreb and Director of the National Centre of Scientific Excellence in Personalised Healthcare. In 2017, he initiated the launch of the Human Glycome project and is one of its two co-directors. His research team is pioneering high-throughput glycomic analysis and the application of Glycan biomarkers in the field of precision medicine. By combining glycomic data with extensive genetic, epigenetic, biochemical and physiological data in a systems biology approach, they are trying to understand the role of glycans in normal physiology and disease. Our next presenter is Michael Butler. Michael is a Distinguished Professor Emeritus of the University of Manitoba in Canada, Adjunct Full Professor in University College Dublin, and presently is engaged as a Principal Investigator in Cell Technology at the National Institute of Bioprocessing Research and Training, NIBT, in Ireland. He holds degrees in Chemistry and Biochemistry from the Universities of Birmingham, London in the UK and Waterloo, Canada, and has held positions as Chief Scientific Officer, NIBT, Associate Dean of Science in Manitoba, and a Scientific Director of MabNet, a Canadian network for mab production. His research work focuses on the development of bioprocessors using mammalian cells for the production of recombinant proteins, monoclonal antibodies, and viral vaccines, an area in which he has co-authored 7 books on mammalian cell technology and over 200 scientific papers. Completing our panel today is James Duffy. James has worked for LabCorp Covance for over 13 years, with his time split over periods in small molecule LC-MS bioanalysis and large molecule biopharmaceutical CMC solutions. In his current role as a manager of a team of R&D scientists within the mass spectrometry development team, James specialises in LC-MS glycan analysis. This includes developing workflows for the characterization of N-glycan profiles for a range of monoclonal antibodies and fusion proteins, as well as characterizing O-glycan via middle-up LC-MS analysis. In addition, James has vast experience in other LC-MS biopharmaceutical characterization methods, such as intact mass, middle-up analysis, and peptide mapping. And finally, our moderator for today's discussion is Mike Claiborne. Mike has worked in the pharmaceutical industry for almost 25 years, principally in drug development and manufacture. He is widely published and currently runs a consultancy business. And so, without further ado, I'll turn the presentation over to Arnaud Delabelle. Arnaud?
Welcome to everyone. I am Arnaud Delabelle, R&D and Innovation Director at Quality Assistance, and I will give this introductory talk entitled Glycan Analysis in Biopharma, Challenges and Future Directions. First of all, very quick words about Quality Assistance. We are a CRO, a contract research organization that provides analytical services to pharma and biopharma companies. We were founded in 1982, 40 years ago, and we are today 240 employees located in a single site in the south of Belgium. We work in compliance with FDA and EMA regulations on all types of products, from small molecules to biologics, but also on cell and gene therapies and mRNA. To know more, feel free to visit our website. Let's focus now on the topic of today, glycosylation, and let's see first why it is often considered as a critical quality attribute of biotherapeutics. First of all, glycosylation is one of the most common post-translational modifications in nature. It is estimated that more than half of all proteins found in nature are glycosylated. But if we consider now therapeutic proteins only, the share of glycoproteins is even higher. A lot of drugs are actually glycoproteins, and that's the case of the vast majority of monoclonal antibodies, such as adalimumab, infliximab, rituximab, or trastuzumab. Antibody-based biotherapeutics, such as antibody-drug conjugates or some multispecific antibodies, are also glycosylated. But it is also the case of other modalities, such as Fc fusion proteins, for example, etanercept and aflibercept, EPO, or granulocyte colony stimulating factor. On the pictures at the bottom of the slide, you find the 5 biotherapeutics that are among the top 10 selling drugs in 2021, and all of them are glycosylated. Let's go back now to the basics. What are glycans? Glycans are made of monosaccharides, the most common of them being presented on this slide. Some of them are neutral, but 2 monosaccharides are charged, and these are the 2 sialic acids, N-acetylnuraminic acid and N-glycolylnuraminic acid. This will confer specific properties to the glycans and thus to the glycoprotein. And these monosaccharides can be linked in different ways, as shown here, which will once again influence the biological properties of the glycoprotein. The N-glycans are linked to an asparagine residue. This asparagine should be part of a consensus sequence, which consists in the asparagine, followed by any amino acid except proline, and a serine or a threonine. The core structure is made of 2 N-acetylglucosamine and 3 mannose residues. The first N-acetylglucosamine can be fucosylated or not. We can then have different classes of N-glycans, such as high mannose, hybrid glycans, or complex glycans, and all these different structures will confer specific properties to the glycoprotein. There are also O-glycans that are linked to serine or threonine residues, and in that case there is no consensus sequence, which means that virtually all serine or threonine residues could be partially glycosylated. The different core structures are presented at the top of the slide, and these structures can be modified, sometimes with sialic acids. A few examples are provided here. The glycosylation of therapeutic proteins will depend on different factors. First, the expression system. It should be chosen such as it produces glycans that positively affect the potency of the drug while avoiding safety issues to the patient. If needed, glycoengineering can be used to optimize the glycosylation profile and thus the physicochemical and biological properties of the drug. And the second main factor influencing glycosylation is the manufacturing process, the cell culture medium, the pH, the temperature, and also the production method, fed batch or perfusion. It's quite clear here that the process in a whole has a critical impact on the glycosylation of the final drug. This glycosylation profile will then impact the properties of the therapeutic protein. First of all, its solubility and stability, as glycans can mask hydrophobic amino acids and improve stability, and more specifically, charged glycans that may improve solubility. They can also stabilize the native structure and improve resistance to denaturation and protect against aggregation, which will improve the safety of the drug. Regarding pharmacokinetics now, here there is a critical role played by two receptors, mannose receptors and ASGPR receptors. The presence of sialic acids will improve serum half-life by masking galactose residues, and on the opposite, proteins containing non-human glycans will be cleared more rapidly. It should also be noted that the glycosylation of small proteins is a way to improve their PK. Glycosylation can also have a role on receptor binding, but in that case it will be receptor dependent. And finally, glycosylation can impact immunogenicity, as some glycans are immunogenic, such as those that contain aglycolyl neuraminic acid, gal-gal motifs, or fucols with alpha-1,3 linkage. On this slide, I have listed the major glycans that will be found on monoclonal antibodies with their impact on the activity. As you can see, some glycans will lower the serum half-life of the protein, such as high mannose. Some will enhance the ADCC activity, such as non-fucosylated antibodies, while galactosylated glycans will improve CDC. Glycans containing terminal sialic acids will have anti-inflammatory properties. And finally, some glycans can be immunogenic, as I said before, such as those containing gal-gal motif or containing N-glycolyl neuraminic acids. I think we have now set the scene for the next part that will focus on glycan analysis. Glycan analysis can be very complex, as there are many ways to characterize the glycosylation of a protein at different levels. This characterization is required by the authorities. For example, ICH Q6b requires the characterization of glycosylation for all therapeutic glycoproteins, and the implementation also of release tests whenever warranted. But this is also the case of other general guidelines from EMA or FDA. This slide presents the different approaches that we use at Quality Assistance for glycan analysis, depending on the type of glycans present, the level of analysis, and the information required. These methods are similar to what is listed in several regulatory texts, such as USP 129 that deals with the analysis of recombinant MABs, USP 1084 on glycoprotein and glycan analysis, or European Pharmacopoeia 2259 on glycan analysis. I won't go into much details, but you can see that the analysis can be done at the protein level, at the oligosaccharide level, as proposed in USP 212, or at the monosaccharide level, as described in USP 210. Of course, you won't have to perform all the tests. It will depend on the information you need, but also on the expected impact of the glycosylation on protein safety and efficacy. I will now give a few examples, starting with the analysis at the glycan level, which is probably, at least for monoclonal antibodies, the most widely used approach, both for characterization and release testing. The workflow is shown here. Over the recent years, with the availability of new commercial labeling reagents, the sample prep time are drastically reduced, and it is now possible to prepare samples in less than an hour. Automation of the process is also possible. Whatever the labeling reagent used, the workflow remains the same. First, a denaturation step, then N-deglycosylation with an enzyme that is most of the time PNGS-F, derivatization with a reagent that will allow both highly sensitive fluorescence and MS detections, and finally purification. These labeled glycans are then separated by LC and detected by fluorescence for quantification and MS for identification. The identification principle can be the following one. First, you inject a dextran ladder in your LC conditions to calibrate your system and convert the retention time into what is called glucose units or GU. Then you inject your sample, and based on the GU value, candidates are proposed for each glycan, and this identification is confirmed by MS data. Databases are now available for different labeling reagents. Here is the fluorescence chromatograms obtained for 3 independent preparations of a monoclonal antibody using helix separation mode. The identifications were automatically given by the processing software, and as you can see, the analysis is highly reproducible as the 3 chromatograms are perfectly overlaid. The ratio of each glycan is calculated based on the relative areas of the different peaks. Here we have the chromatogram of a more complex sample, etanercept, which is a fusion protein with more complex N-glycosylation. But once again, glycans are identified unambiguously and automatically. The advantage of using instant dyes is the MS sensitivity. These dyes were designed to allow quick labeling, but also high sensitivity in both fluorescence and MS. The arrow here shows a glycan with 0.6% of relative intensity. Here is the MS spectrum obtained on a state-of-the-art high-resolution MS system with 2AB labeling. Not bad, but quite noisy. And with the recent dye, the intensity is much higher, which allows for better MS identification of low-abundant glycans. Other separation modes can be used, such as mixed mode, here ion exchange and reverse phase, and in that case, glycans can be separated based on their charge, with first a massif with neutral glycans, monosialylated, desialylated, and so on, which is very useful for highly sialylated glycoproteins, such as phetuin or etanercept. Using another mixed mode column, here ion exchange and helic, a simpler separation between the different charges is obtained, and this method is perfectly suited for QC analysis of sialylated proteins. For O-glycans, the approach is much more complex. First of all, there is no enzyme that is universal for O-glycan release. Chemical release can be used, but it has to be optimized. In the example I will show, we used alkaline beta elimination under reducing conditions, which led to limited peeling side reaction. We did not label the glycans, and we separated them on a PGC column. And finally, detection was done by MS in negative ionization mode. Here is what we obtained for etanercept and phetuin, which allowed us to quantify and identify different O-glycans based first on MS data, but also fragmentation data to confirm the structure of the glycans. Another approach is also to work at the glycopeptide level, which can be very useful to perform site-specific glycosylation analysis. In a regular peptide mapping using reversed phase separations, glycopeptides are lost in the middle of non-glycosylated peptides, as you can see here on this chromatogram. In that case, you need MS to identify and characterize them. If you use high-energy MS, fragments that are specific to glycopeptides can be identified, but as you can see, on the total ion chromatogram, they are much less easy to detect. Another approach is to use helix separation. Here, we analyze the model compound RNase B before and after deglycosylation. You can easily see that the glycosylated peptides are well separated from the non-glycosylated ones. And when you zoom in on glycosylated peptides, you can see excellent separation between the different glycoforms. Once again, this is a great alternative to reverse phase separation. And I will now finish with the analysis at the subunit level, which is a more recent approach, but also offers a lot of advantages. You probably know the enzyme IDES, commercialized by Genovis under the trade name Fabricator, but there is also the IDZ enzyme that has an improved activity for IgG2A and IgG3 mouse antibodies. Both cleave the antibodies below the hinge region to yield Fab and Fc fragments, and after reduction, you obtain 3 subunits of around 25 kilodaltons. With this relatively low molecular mass, it is possible to get high-quality chromatographic separations, but also good MS resolution and accuracy. If you use helix separation, you get a very good separation between the 3 subunits, but also for the Fc fragment, an excellent separation between the different glycoforms as it is shown here. Quite interestingly, we showed on a therapeutic antibody that the different approaches that can be used gave very good agreement for the quantification of the main glycoforms G0F, G1F, and G2F. Even on complex glycoproteins, such as etanercept, that is both N and O glycosylated, this approach can be used. Here, the sample was N-deglycosylated to analyze specifically O glycans and then digesting using IDES. We obtain a very broad peak for the TNF receptor subunit as seen on the right of the chromatogram, but by analyzing the peak in small fractions, the composition in O glycans can be obtained as shown here on the different mass spectra that correspond to different fractions of the peak. And to conclude, I will talk about glycomics approaches that are not used much today in biopharma, but that can be very useful anyway. The idea is to use methods that are commonly used in biopharma, but to automate the sample preparation to make high-throughput analysis possible. In these studies, samples are sometimes complex, and we sometimes have to analyze blood samples. Some steps of the preparation can be automated, such as deglycosylation, labeling, or SPE purification, and then the samples can be analyzed by different methods, such as LC or CE, with fluorescence detection, MALDI-MS, or LCMS. The applications of high-throughput glycomics are epidemiological studies, genetic or clinical studies, or diagnostic applications. This approach can also be useful in early development for the glycoprofiling of biopharmaceuticals, as I have shown before, when you have many samples to screen or during the manufacturing processes in order to optimize it. But high-throughput glycomics can also have another important role in biopharmaceuticals development. Glycans are indeed responsible for a large part of inter-individual variations, and biologicals may have very different activity depending on the glycocontext of a patient. To take an example, therapeutic immunoglobulins compete for Fc receptors with immunoglobulins of the patient. And in this context, a patient with 20% of afucosylated IgGs that bind quite strongly to Fc receptors will be very different from a patient with only 2% of afucosylated IgGs. High-throughput glycomics can therefore help to better understand the inter-individual variations of the efficacy of a treatment by analyzing the glycome of the patients. That's all for this presentation. It was quite quick, and of course we did not have time to cover all the details of glycan analysis, but if you want to have more details, you will find more information in these different publications that are shown here in this slide. It's now time to switch to the expert discussion to go deeper into the details of glycan analysis with the associated challenges and the future directions.
Okay, thank you, Arnaud, for your excellent presentation. This sets the scene well for our discussion. And I'd like to start off the discussion with a couple of questions about your presentation. You've described glycosylation as a critical quality attribute and have broken that down into a complex map of analyses of a range of glycan characteristics. How do you package the multi-attribute data that you generate to understand your critical quality attribute and establish acceptance criteria both for in-process and for your products?
Yeah. So as you said, the analysis of glycan is very complex. There are a lot of tests that can be done at different levels. You can study the sialic acid, the N-glycans, the O-glycans, but the analytical part is only one thing. You have to know your manufacturing process very well also to in order to better understand the impact of the manufacturing process on your glycosylation, the variability you may have. And then that's only when you know also the manufacturing process and the activity of your product, because glycosylation may have an impact on activity or may not sometimes. Once you know the variability of your process and the impact of glycosylation on the activity of your product, then you can begin to establish acceptance criteria. At the beginning of the development of the product, these acceptance criteria may be quite broad. And then once you get more insight, more information about the activity during the preclinical, clinical studies also, then you can start to have narrower acceptance criteria and then continue working on your product.
Okay, thank you. Can I just open that out to the panel, please? Would anybody else like to make a comment about the complexity of this and how you establish acceptance criteria? Can I bring you in, perhaps, James? Would you like to make a comment?
Well, that's pretty much exactly what I would do as well. Obviously, we'll look at the product we're working on initially, address all the possible CQAs where glycosylation may come into that, in terms of can it impact its mechanism of action. Highlight all these key areas in terms of the glycan specifically. Again, if we're doing a release glycan assay, it would be then grouping these particular glycans together, such as in afucosylation or terminal galactose, if either of those would be impacting the mechanism of action, and then highlighting that and then addressing that in terms of reproducibly replicating that overall abundance, again, starting wider initially, but then, as Arnaud said, bringing it in gradually towards the end of the process to have a more robust assay. So yeah, I agree effectively.
Okay. Well, thanks for that. Gordon, Mike, would you like to add anything to that? There seems to be a consensus there. Would you like, Gordon, do you want to come in?
Yeah, so I can add a little bit to that. So we have two problems here. First, we don't really understand how glycosylation is being regulated, both biologically and in the production. So it's not easy to tightly control the product. And the second thing is, in majority of cases, we don't know what glycans actually do. And in a more complex proteins, it can be glycosylation site specific. So if we look at the general galactosylation level, it might not be so relevant, but at the specific glycosylation site, it could be. For example, for antibodies, FAB and FC glycosylation have very different functions. So I think it is essential to, with time, develop knowledge about the functional aspects of individual glycosylation feature, and then adjust the regulation in a way that something is tightly controlled, and other aspects do not have to be so tightly controlled, because if they don't affect the functional properties of a molecule, it is not so important. But if they are affecting the functional properties of a molecule, then it is essential.
Okay, thank you, Gordon. Can I bring in Mike there? Would you like to add anything?
Yeah, I mean, I think that was well said, insofar as I think there needs to be a lot of structure to function analysis. And the more of that type of analysis that can be done, the more we will understand what structures affect certain physiological properties, what affect the therapeutic properties. So I think it's more knowledge there be essential moving forward.
Okay. Great, thanks. I'm going to move on now to the next question, if I may, and that's again to you, Arnaud, with regard to your presentation. And in your presentation, you brought in the regulatory guidelines into frame, which is obviously very important if you're developing products. And given that the design space around the analysis of all these glycan characteristics is complex, we may also be talking about novel glycans. Are the regulatory challenges significant in delivering a commercial product as an endpoint? Another way of putting this, are the regulatory guidances we have fit for purpose? So if you could respond to that, Arnaud, that would be wonderful.
Yeah, so I listed a few chapters of the Pharmacopoeia of the ICH that are talking about glycosylation and how they should be regulated. But if you have a look to these guidelines, especially the more general ones, they say that you have to analyze the glycosylation, you have to study the impact on the activity, but you don't have much detail. So you are quite free to work on your own product and better understand it. So I would not say that the regulatory guidelines are really strict. They are just a guide to know what you have to do. And then the chapters in the Pharmacopoeia describe some methods that you may use, but you don't have to use these ones if it is not strictly required by the monography in the Pharmacopoeia. So you are free, but there is a lot of work to do. And for some products, it may help to have guidelines that are maybe more strict, more precise about what you have to do in order to put a product to the market. But once again, even if we consider an antibody, all antibodies won't have the glycosylation won't have the same role on all the antibodies, depending on their mode of action. So it's not easy to have something that would be fit for purpose for any product that you want to put on the market.
Okay, thank you. I'm going to bring Gordon in now just to add a little bit to this story. Gordon, would you like to make a comment?
Yeah, so I think we have to be aware that the glycoengineering is still its very infancy. And we have to be careful not to kill the field by going too strictly at the beginning. And especially because manufacturing is still an issue. So it's not easy to replicate a glycosylation profile if you move from bioreactor to bioreactor and so on. So I think with regulation, we have to move forward with time and bring stricter and stricter regulation for important things, but not move too quickly to prevent any kind of modifications, because we have to be aware that this is technically difficult. It's not easy to do. It's analytically challenging. There are not many laboratories in the world which can do proper glycoprofiling. So we have to move with a pace which is acceptable to the industry.
Okay, thank you. Mike James, would you like to add anything to that? Are you happy for us to move on?
Yeah, I mean, a comment that I would make, I think Arno made it earlier, is that, the glycan functions in different ways, and where we have different therapeutic modalities for different antibodies, we might need different glycan attributes. And so we're looking at different things for different functions, and that's an important aspect.
Okay, thank you.
Just on that as well. Yeah, just again, I think we have to go with the times, especially when you're thinking of MABs. You're thinking your glycan profile is relatively simple, straightforward. A lot of the products I work in characterization of fusion proteins, and some of the profiles I'm seeing in terms of the glycosylation and the complex and maturity of the glycans, it's really vast compared when you compare that to a simple MAB FC glycan profile. So I can imagine that to try and restrict the regulations or guide the regulations at this point stringently is going to be very difficult. So I think a very flexible approach would be the most suitable way at the current time.
Okay, that's a point well said. Okay, I'm going to move on to the next question, and I'd like to bring in James to address measurement capability across that complex analytical map that we've seen described. And the question is, in order to get the sensitivity and selectivity needed for the different elements in that analysis workflow, that complex analysis workflow that's been mapped, could you expand a little bit on where we are right now in terms of analytical capability, and that could be hardware or software, and where the gaps are in that capability currently?
Okay. Well, from my perspective, in order to achieve the desired mass spec sensitivity for the identification of glycans down to the relative abundance of 0.01%, that's kind of where I want to work. I would always use glycan label with the tertiary amine, because the 2AB is just not going to cut it in terms of the identification that I need specifically for the products I'm working with. In terms of selectivity, if we're trying to think of gradient separation, we can tweak that to pull out a specific glycan of interest. In terms of the software I'm working with, I'm relatively happy for what we need to do. The issues are coming is when we're going to transition from where I'm working in the kind of the non-reg space in terms of characterization, new molecules, is going into a QC environment and batch release, which is our end goal. It's a LabCorp drug development. But the main issues are going to be the software and the capabilities of automation of that. So a lot of the software I use, and I think a lot of the other softwares, they always go with a first pass identification of GU value, using GU value ahead of mass spec, and this can lead to several misassignments, which ultimately leads to manual alteration of the data, and thus impacting the resulting report functions as well, making it more difficult to get a standalone report. So if you want to do high throughput or potentially pass this down to a non-glycan expert within our QC lab, it makes it a little bit difficult with the current applications in the software for that kind of batch release approach of glycan analysis.
Yeah. Okay, thank you. Could I ask you, Mike, perhaps if you can, if your experience in terms of the capabilities we have currently, how do you feel things are at the moment?
Well, I mean, the type of analytic tools that we have, I mean, a lot is developed from chromatography, HPLC, UPLC, with the different types of columns and so on, and relating to orthogonal techniques using mass spectrometry. Those are the tools that we have, and those tools have slowly been improved over time. So I think I have nothing further to add than that. And I think, there is a question later on about there are introduced the databases that are available. They're, of course, very important reference databases that are available online to be able to relate data in individual labs to a consensus data, which is provided globally.
Okay. Thank you. Gordon, Anna, would you like to add anything to that in terms of the capabilities that you have and what you might like to see in the future?
Yes. So I agree with what James said, and we also observe some problems with misassignments when we use the identification workflow based on GU values and mass spec confirmation. It would be nice, I think, in future versions of software to have first better databases to make sure that we can identify even some rare glycans that are not the main ones we find commonly on antibodies, but also to use maybe MSMS data, fragmentation data, in order to better identify the glycans and confirm exactly the sequence.
Okay, thank you. And final word to you, Gordon, if you wish to add anything further to that.
Yes, we do have a problem with data analysis. So recent comparison of the different labs where the raw data was sent to their different labs generated very different interpretation of the same data, which is concerning. So the problem is that when you compare the results from different labs, there can be very different interpretation of the same data. So obviously this field needs more work, needs some more people working in the field. And I think the machines are getting better and better. So really we can get beautiful data from the new machines, but the complexity of a Glycome requires more effort in developing software tools to actually see what we have in the data.
Okay. Well, if we can perhaps move on to the next question and build on that point you've just made around the informatics side, and perhaps whether commercial versus more tailored in-house software is where we're at right now. Could I ask you, Michael, the question is around this generation of a lot of multi-attribute data, and obviously an informatics system is needed to support that analysis. For example, how do you achieve a detailed analysis of your glycan pool using glycoinformatics, and then are the commercial tools adequate? How do you approach the analysis then of things like novel glycans?
Yeah, I mean, I think it's good. This question relates to the previous question. I mean, we certainly need an improved databases. I mean, these are databases which involve collection of data from different labs, and that's the only way we can get a sort of consensus of analytical methods. The type of thing that I'm talking about at the moment, there is available GlycoStore, which is a database with a unique collection of, when I last looked, is about 850 unique glycan structures. So these are annotated through chromatography, through helic chromatography, reverse phase, capillary electrophoresis, and mass spec data. So, if somebody is analyzing, a lab is analyzing a novel glycan or a novel protein or whatever, I would suggest the first port of call is to compare the data that is generated from the instrumentation in that lab with such a database like GlycoStore. That would give some confidence, I suppose, in interpreting the data and assigning structures. Obviously, it's more difficult if you're assigning a novel structure, a structure that hasn't been seen before. That type of protocol is not perfect. We certainly need to improve on that. There's always a need for improvement in analytical tools, analytical methods, and so on. But that's, I think, a good way of moving forward to ensure that these databases, there may be others too, but I'm using GlycoStore as a good example of one that should be replenished and should be improved gradually over time, and then we can rely more on those in terms of interpreting the novel data that we gain from our analyses.
Okay. Can I ask you, James, then, where are you at now with commercial versus bespoke software to help with your interpretation?
So I'll be honest, we've worked with a limited library for a long time, and I've basically made a homemade library based on all the products I've been working with. And again, each time that is just looking through the masses we're seeing, detecting that where possible trends appear in terms of, if you've seen a glycan earlier, will you see a similar glycan with multiple more antennae or silation? That's how I've come out, and obviously I've listed those. So every time I go back to a similar products, I'm using my homemade library to do that. In terms of novel glycans, what we've also done when of particular interest and on either innovation work we're working at for fusion proteins or client work as well, as potentially adds exoglycosidase digestion to add further confidence in the assignments. So then this is just simply cleaving back the different residues and seeing if we see the same conversion to the expected glycans at the same relative abundance. That gives us more confidence in and clarification in our assignments that we have initially made, to then see if we can see them be truncated back to the subsequent converted glycans following enzymatic digestion of silidase, galactosidase, and such. And that's really helped us gain confidence in a lot of our library. And then when we see the similar mass appear again, we can have more further confidence that we don't necessarily have to do the exoglycosidase digestion on that product, but we've got good basis to identify with that species.
Okay, maybe I can ask you, Gordon, is there any collaboration going on across all these wonderful experts we have in this field that to help generate the right sort of databases that are needed?
Yes, of course. But before that, there's one important aspect which is important to emphasize, and that in addition to databases, we need chemical standards. Because if you take the same sample and just run it through the different instruments in the same lab and then go back to the database, often you get the different assignments. So obviously the databases are highly lab and instrument linked. So what we are doing at the moment, there is an initiative called the Human Glycome Project, which one of the goals is to generate this type of standards. This is a line of work led by the Rick Cummings at Harvard. So the idea is to put in a small vials every single glycoprotein there is in a human body and make it available to researchers. I think this would be one very important achievement. Of course, this would be human specific, but most of us are human focused. And the second thing is there is a try through the Human Glycome Project to integrate the bioinformatic pipelines, which is still extremely challenging. We haven't moved far, but at least we are trying. So I think the Human Glycome Project is one of the good ways to integrate all the activities. And my feeling is since I'm on the interface of academic and the biopharma, that we need more interaction between academia and biopharma.
Right.
Because these things go in parallel, and I think we need to collaborate more.
Okay, thanks. And a final word to you, Arno, on this topic. Would you like to make a comment?
Yeah, I agree with what was said. One point maybe regarding the software. In-house software are good, but we have to keep in mind that in a biopharma environment, in a regulated environment, that is not something that is easily applicable. So we need also commercial software that can be applied in a regulated environment and that can work correctly and identify correctly the different glycans for release testing and regulated characterization.
Okay, thank you. Okay, I'm going to move on to the next question, if I may. This is a question initially directed at Gordon. This is really about the practicalities of streamlining the glycan analysis from sample preparation to data analysis and output. And it's a question about high throughput procedures, and really I'd like to get a view from you, Gordon. What currently is the state of the art in high throughput procedures in the area of glycomics and glycan analysis?
So this is actually a very complex question, and I think the only solution eventually will be to stop treating all glycoproteins as equal. Because if you think about proteins, we do not analyze the glycated hemoglobin, liver enzymes, and some complex membrane transporters in the same way. Also, it is very different if the sample comes from a cell culture supernatant or human plasma or something else. And eventually, we will have to streamline kits and procedures for each of these cases. So we can have a general glycan kit, we'll have a kit for IgG or whatever. And we have to acknowledge that the glycoproteome is several orders of magnitude more complex than the proteome, and we have to address it accordingly.
Okay.
And of course, data analysis is very important because, as I already mentioned, using the same raw data, different labs generated different output, which is not good.
Yeah, okay. Can I just go into a little bit more detail about that in the area of a critical area of sampling from, say, a bioreactor and sample preparation? That's the initial stage of doing your analysis. First of all, is that a bottleneck in trying to establish high throughput? And then what do you see as the current need in both sampling and sample preparation capability to support glycan analysis?
So usually the biological sample is very complex.
Yeah.
And we have to somehow enrich for what we are looking at. And this is definitely a large bottleneck, because if our sample is not homogeneous in a way it is being sampled and enriched, then actually this can be the source of variation. And in our hands, when we do high throughput, it's usually the protein purification or enrichment, which is the trickiest part. Because once we get to glycans, we have streamlined the high throughput procedures, it's not a problem. But to get a pure protein, it is a problem. So this is something what we'll have all work together to develop good kits, good procedures for sample prep.
Yeah, okay. James, can I bring you in there? You've got your hands on very much in method development. What's your view on where we're at now with high throughput, and what do you need?
Again, we could do a lot of samples in the lab. There's plenty of new automation kits in terms of that can speed up the time in the lab, but I don't see that as a downside. I don't think it takes the lab time, as I think I would mention in the presentation, you can do it within 30 minutes. It's not the problem. It turns out it's the processing of it, and depending on the samples you're looking at the ends, it's the length of time it takes to go through and stringently go through your glycan profile. If you're looking at potentially early stage glycans, you're just looking to sell it, and if you're trying to just differentiate between a lot of samples, I would recommend looking at maybe subunit analysis. Again, if this was a map, I would look at the FC only, get a reasonable idea of what's going on in your glycan profile. Maybe it's the most abundant 6 or 7 glycans. and then that would guide you significantly enough to basically limit a lot of your samples initially, and then that saves so much time in terms of the data processing side of things instead of doing a full release glycan. And then you basically would narrow it down and then do full release glycan analysis on a handful of candidates. That's how I would work through that process.
So the smart approach rather than trying to do everything.
Well, yes, yeah, work smarter, not harder. Yeah.
Okay. That sounds good to me. Arno, do you want to come in there at all on on where we're at with high throughput? Is that maybe an important aspect of your job?
To be honest, it's not a very important aspect in my activity. It can be in other activities, but we don't have many samples to analyze. So the quality at the end, the quality of the data is more important. But anyway, automation can be useful to save time in the lab, but also to get more reproducible sample prep, and so more reproducible results at the end. So we don't use large robots, but small automation devices can be quite useful in order to optimize the sample preparation step in order to get better quality data at the end.
Okay. And a final comment to you, Mike, on this topic, if you wish to make any.
Yeah, I agree with everything that's been said. I mean, the only comment that I'd make, of course, with the glycan analysis, there are differing levels of complexity that you can undertake for the analysis. And it's important to consider what you're trying to achieve with the analysis. So if you're just looking at monitoring a bioprocess and take time samples, then, maybe the level of analysis that you want is not so detailed, just because analytical tools and analysis is available doesn't mean to say that you have to use them fully on every occasion. You just have to use them in a way that addresses the question that you've posed or the problem that you're looking at.
Okay, great. Thanks, Mike. I'd like to stay with you, Mike, if I may, for the next question, just drilling down into a little bit more detail here. And this is about specific types of glycans. What currently are the analytical challenges, in your view, for structure elucidation of N-glycan isomers? How critical is that?
Yeah, isomers are very interesting because you have, a degree of complexity with the glycans that not only have you these subunits that come together in terms of proteins, they fit together in a common way. In the case of glycans, individual monosaccharides can bind and link in different ways, and that's quite important to both structure and function. And the problem there is that if you go through looking at masses, then the mass is the same. So if you look at the parent ion, certainly in mass spectrometry, you get the same value. So the approach that we've used in several occasions is to rely on specific enzymes. The specificity of enzymes is quite important in terms of, deciding what you have, whether, for instance, you might be looking at linkages of sialic acid, might be 2,6 or 2,3 sialic acid groups. The molecular weight's the same, but the linkage of the sialic acid is different. So what we have used in those cases are specific siladases, which we'll know will break at certain linkages, and that's proved quite useful in determining, and it is a problem with sialic acid. We know that if you're trying to form a human glycoprotein, you need the 2,6 linkage, whereas from CHO cells, hamster-based, or even murine cells, you get a lot of 2,3, a different sort of linkage. So you need the appropriate siladase to be able to break and understand, how the activity is working. So yeah, isomers are important, and it is there are different ways. We've approached it from the enzymatic point of view, breaking down in different ways. There are, of course, different ways in mass spectrometry by fragmentation patterns of the same parent ion that you can go through. So it is something that's slowly been advanced, and slowly we're understanding more and more.
Okay. Thanks. Thank you, Mike. If we can treat that as a comprehensive answer to that particular problem, and just in the interest of time, I'd like to move on to the next question. And this one is directed at James. Arno, in his presentation, mentioned O-glycans. And what the question is, with the current lack of suitable release glycan methods for O-glycans, what are the most appropriate ways to characterize O-glycan population? Pastor James.
Thank you. Again, the dream is that somebody fathoms out a release technique for glycans, and that's going to be the dream at some point. But until then, the only way I've managed to work it is, again, depending on the product. Again, you can have multiple sites of all glycans and makes it much more complicated. But again, would be used a middle of approach. Basically, getting the product down to smaller subunits, and then removing any end glycans that are present to simplify it, and then kind of truncating back with, again, sialidases and other exoglycosidase enzymes to truncate it to its intact mass, and then build it back up again, effectively the same way. So try and get it down to nothing, and then build it up sequentially back to where we believe these glycans are. And then using that information, we can then link it to when we know what roughly the glycans will be or what residues are included. We can then use that data to imply that to our PEP map analysis, and then treat it again. I think Anaud went into a little bit of detail, but that would be our approach as well. Again, try and get as much information as we can at the subunit level by truncating back and then building back up those glycans after removal of the end glycans, and then looking at the peptide level at the specific serines and threonine sites with the potential and then looking at the masses we detect at that level.
Sure. Can I bring you in, Arno, since you raised this as a particular issue in your presentation, and what's your views on trying to define a specific method, or are we still stuck with the approaches that James has described?
Yes, so I agree with James. Working at subunit level can be useful, at least to start with, and see what you have in your sample. There are some ways for chemical release of the O-glycans. It's not easy, but it can work. But what I want to mention is that the amount of information you need for O-glycans will depend also on their influence on the activity of the protein. Sometimes the influence is quite limited, and in that case, you can limit your analysis to high-level views such as subunit. Of course, if you see that some O-glycans, especially some sites, have a critical impact on the activity, on the stability, on the safety of the protein, in that case, you need to go further, but it will be a case-by-case approach.
Okay. And Gordon, Mike, would you like to add any further to that on the O-glycans?
Nothing further from me. I agree with what's been said, and that's
I'm
fine.
sure. Yes. Okay. Well, if we don't need to say any further on that, I think we've answered that question, and we don't need to go into any further. So I'm going to go on to the next question, and this I'm afraid is you again, Mike. This is about labeling. So labeling of released glycans, for example, to characterize the glycan pool is obviously an important step, and we've heard that several times now. What are the advantages and disadvantages of the different labels we have in use that for glycan analysis?
Yeah, there are different labels that are commercially available now. And, it's interesting to look back on the history of this. I mean, 2-aminobenzamide, 2AB, has been a label that's been used extensively, particularly for chromatography, HPLC, UPLC, where you're looking at a fluorescent tag on the glycan. and the advantage of there've been a lot of work on that is that there are good databases on 2AB. A lot of the work was done by the group in Oxford, and they developed an awful range of databases. Since then, there have become available what are often termed the rapid tags, and they work on a slightly different chemistry insofar as they are glycosylamine attachments through NHS and hydroxysuccinimide. And they address several questions, really, and what is the ideal tag? Well, you want high fluorescence. 2AB is fluorescent, but can we get higher than that? And it's true. If you can increase the fluorescence of some of these rapid tags. We want them to be mass spectrometry compatible. So this is where the tertiary amines that were mentioned before, I think by James, are important in being compatible and being observable with mass spec. And then we want, and this has become a question, functional exoglycosidase activity, whereas exoglycosidase arrays are commonly used where you break down the glycan sequentially. They can be used fairly well with 2AB when you get these rapid tags. The increased molecular size sometimes have stoichiometric effects of preventing exoglycosidase activity. So we've got to be conscious of that. And the fourth thing is database availability. I mentioned earlier the 2AB. There are good databases available. Whenever we have a new label, it's important to realize the GU values of the labeled glycans are going to be different. So we need to produce new databases. We actually have been working hard on a database for instant PC, which is one of these rapid tags. So it's important that along with the advantages of the rapid tags, if you like, then we need more information of how they work and how we can interpret data from them.
Okay, thanks. Arno, do you want to come in there? In your laboratory, how are you managing this labeling issue? And
yeah, I fully agree with what Mike said. The instant tags were quite useful for us in order to improve the throughput of the analysis. As I said, the throughput is not critical, but if you can spend 1 hour to prepare your samples or even less instead of 2 days, of course, it will be very interesting. And these instant tags also added the sensitivity both in fluorescence and MS. Yes, and finally, clearly the availability of the databases and the quality of the databases that are available are critical for the identification.
Okay, super. I got Gordon.
So
would you like to add anything to that?
I can add a bit to that. So 2AB is not very good dye, but we still mostly use it because we have 200,000 samples analyzed in our lab using that dye, so we can directly compare to that. Just last week, we published a paper comparing, I think, 9 or 10 different dyes, and they're all different. Some of them are way better than the 2AB, but also it's instrument specific. So dyes do not behave the same in the different instruments. So there's definitely need for more work. There are new dyes. Several producers offered these rapid dyes, more mass spec friendly dyes, but I'm not absolutely positive that this is the optimal solution. And eventually we'll have to come to a standard and then everybody stick to the same standard, because otherwise we'll have many overlapping databases and having conflicting information. So I think it's really important to focus on a dyes, make some kind of industry standard, and then let's try to stick to it.
Okay, super, thanks. I'd like to leave that question there and then move on to the next one. And this is directed at you, James, if I may. Arno in his presentation mentioned the importance of sialic acid, and I think that topic's been raised a few times in the responses that we've heard. James, can you comment on the importance of sialic acid content as a measure and techniques to analyze this? In a sense, in a way to complement the other measures that we've been discussing.
Yeah, so it's one of my biggest concerns when I'm characterizing an N glycan population. It's to ensure that we are detecting any galactosialic acids and also alpha gal as well, alpha galactose. Mainly because of the immune reactions that can be causing patients even at low abundance, and again at low abundance being the critical thing here. When I've measured these glycans before, I'm always seeing them, individually at least below the 1% abundance level. Again, and this I think would rule out any sort of measurement of this at the peptide level. You're going to have to concentrate solely on the released glycan analysis to identify these. And again, with the mass difference between, if you're comparing the two sialic acids, so that Nana and Naga, as I call it, they're 16 Daltons apart at 1 oxygen. So we should be able to differentiate in mass between those. Comfortably. The problem I see, and that arises when I is the complication of the profiles when you've got multiple co-lutein species and an isobaric species as well. On top of that, when you've got fusion proteins, again, these products I'm seeing these on fusion proteins normally found within the receptor region. Again, what I've mentioned before about in terms of exoglycosides, but there was a specific example I had is when I found a mass which was M over Z 1349, and there was 2 potential assignments to this, and it was one which was G2FS1 with and then plus a galactosialic acid, and then G2S2 with an alpha gal. And to differentiate between these, we used the sialidase enzymes again, and we saw the conversion. Again, this was a relative low abundance, I think 0.5% relative abundance. And we saw, the conversion of this species to a previously unseen species of G2F plus alpha gal, which identified it as an alpha galactose species, not a sialidase, allowing us to have that identification. So that's one way. Again, with the mass shift being as it is, I would expect, mass spec to be the first protocol. If for whatever reason that you do want to then clarify this, we do have a non-mass spec method where we release the sialic acids with either sialidase or a mild acid hydrolysis, and then we confirm the ID and relative amounts of each sialic acid individually. So if you're specifically looking at the different sialic acids and differentiating between those as a standalone method, we have that. And again, this can be used. This can be done just with a reverse phase method on a UPLC with fluorescence detection. And that's another approach we can do when we're confirming our mass data and complementing that, I think.
Yeah. Okay, thanks. Gordon, do you have any comments about this, the sialic acid measurement issue at all?
So it is very important to measure. It can be very complicated if you look at larger structures. So it's challenging, but I completely agree. Yeah, this would be an approach to do it.
Okay, thanks for that. I'm actually going to move on to the next question because James actually raised the issue of alpha gal, and if I can direct that to Mike, we know that alpha gal is an immunogenic glycan epitope. Really, the question is, how do you control that type of entity, particularly in manufacturing? How do you monitor it and control it?
Yeah, I mean, just some background. The 1,3-galactosyl transferase is an enzyme that can be produced in murine cells. I mean, it's the enzyme and the gene is present in most mammals, but not in humans. And it's important to prevent its formation because it's immunogenic. I mean, it's a gal-gal linkage between two galactosyl groups. So what is important there is to realize that if you're using murine cells like NS0 cells, then it's likely that this will be produced. In some of our studies, we found that for instance, 7% of the gal from NS0 cells can be, gal-gal produced. So it's important to realize that, you might want to look at the cells that you're using, and it's the disadvantage of using, cells that have this gene present. That's important.
That's super. Okay, so I'll move on to the next question. I'm going to come back to you again, James. With many therapeutic products, and you mentioned the FC fusion proteins, they have multiple sites for of N-glycosylation. What are the best ways to obtain site-specific glycan information? Because we know that's an important characteristic that we may need to measure.
Yeah. Again, I think, and I touched on this in the presentation, PepMap would be a really good way to get site-specific analysis. And additionally, glycan occupancy at specific sites in case you're seeing a peptide with no glycan present, then you could calculate the level of glycan occupancy at the site as well. The issue being, though, that you'll still only get limited glycan information. And again, this will be of the most abundant species. Again, top 5-6 glycans. What we've looked at, in particularly since you mentioned FC fusion proteins, is a way to physically separate the FC and receptor regions through IDES. The particular FC we were looking at had the papal G amino acid sequence to allow the cleavage to separate those two, and obviously we'll have FC glycans, and then for this we had 8 sites within the receptor region of N-link glycosylation. So once we had the physical separation of the two subunits, we then used protein A to bind the FC and wash off the receptor, thus giving us a receptor-only sample. And then we would add a few more salt washes to purify the FC, and then elute that with some separate buffer and have a separate FC sample. And then we would just run our routine release glycan analysis, and then we would have effectively an FC-only sample, so glycans associated with the FC of that fusion protein, and then the receptor region as well. And this would be a particular use and was of particular importance when we're looking at glycans impacting FC effector functions. So, for example, if you were looking at, you did a full profile, the full release glycan of your full profile, the full product of the FC fusion protein, that this was utilizing ADC within its mechanism of action, ADCC, sorry, within its mechanism of action, and you found very high levels of glycosylation, you would think this would be a positive thing. However, then you could perform this analysis and then identify exactly where those high levels of glycosylation were located, where they are attributed to the receptor region or the FC region, and then obviously that would have huge impacts in terms of the product's ADC levels depending on where they were located. So that was a unique way that we found to not only have idea of everything through the PepMap, but a low abundance, but then specifically getting an FC versus receptor region. And again, it'll be interesting going forward and see how we can link the different areas of glycans to the specific motifs.
Sure. Would you like to add to that, Arno, in terms of how important this characteristic is?
This is very important because depending on which site is glycosylated, which glycoforms, the activity can be quite different. But once again, it depends on the level of information you need, the amount of information. If you only want to identify the main glycoforms, in that case, working at the subunit level or glycopeptide level can be enough. Of course, if you want to detect the minor glycoforms at each site, in that case, you may have to use quite complex sample preparation methods in order to isolate the different subunits or glycopeptides, and then perform a full N-glycan release on these different subunits. So once again, it depends on the information you want, but clearly this can be quite critical for some specific modalities.
Okay, thank you. Gordon, Mike, would you like to add anything further to that?
I would just like, sorry.
Go on.
I would just like to say it's another example of needing information about structure to function, and that structure to function information might be different for different fusion proteins. So it's important within getting that information, we determine what the critical quality attributes that we want are.
Okay, thanks. And Gordon, last word on this topic.
I would just like to add, as we try to move to more high throughput analysis, all the purification steps become really challenging because you can't control each of it, and then there can be a contamination. So I think glycopeptide profiling would be the optimal approach here, but with a LC separation of peptides and then mass spec. So but it is a challenging. So we do have several high throughput methods for very complex proteins like alpha-1 glycoprotein, but it's a long method development to actually have the method which is reliable.
Okay, thank you. I'm going to stay with you, Gordon, for the next question, because it does build on this high throughput theme that has pervaded some of the questions we've had. And really, it's about the important factors in high throughput glycomic studies, what those important factors are. And then the follow-up for that would be, in achieving a quality outcome, is there an important question in trying to balance precision versus throughput?
Okay, so let's first address precision versus throughput. I think precision cannot be sacrificed for throughput. And I think the reason why Genus is so successful in high throughput studies, and we analyze something like 80% of the total output of high throughput glycomes, is that we never sacrifice precision, because we also have to think from the side of people who are giving us samples, and we mostly do human cohorts and the clinical studies. These samples are extremely precious. They cannot give us 1,000 samples if 100 will be enough with a more accurate method. So precision is essential. Regarding automation, as we do a lot of analysis, so approximately we do 1,000 samples a week, which is a lot for the comics. And we do try to automate as much as possible. But so far, our experience is that at the moment, humans are more precise than robots. So the measurement error for our automated procedure, and we have automation either with the small machines or fully automated procedure for IgG, for example, on our Glycan, the measurement error is actually still larger. So the automation needs more development to bring it to the level that it actually generates more precise data than our analysts.
Okay, thank you. Arno, would you like to add anything to that? Your experience with precision, in particular precision versus throughput in a high throughput environment?
Yes, so as I said, we don't work a lot with high throughput, but clearly the precision is more critical, I think. It's better to have a precise method that may be quite slow instead of having a very high throughput method, but that gives results that you can't rely on. So both are critical, but the performance, the precision is critical.
Okay, thanks. So I think I'll leave that question there and move on to the next question in the interest of time. And if I can stay with you, Arno, and this is more in relation to manufacturing and perhaps a little bit of regulatory. In general terms, what does a manufacturing control strategy for glycosylation look like, in particular in relation to the questions around the control of the glycon profile during bioproduction and testing for batch release of clinical product?
Yeah. So it was partly discussed already, but the manufacturing process in terms of glycosylation is something that is not easy to control, and it's very difficult to have always exactly the same product with a very fixed amount of glycans and of the different glycoforms. In the past years, with the first biosimilars, we have begun to see some studies that were published that compared different batches of the same product that were produced over several years at different locations. And we saw that the variability for some of the products for monoclonal antibodies could be quite high, but without any significant impact on the activity, because all the products passed their specifications for the potency and so on. So it's not easy to control very strictly the manufacturing process, even if it's something that we would like to do. But at the end, what is the most important is the activity. And to answer your question about the batch release and the specifications should not be too tight to take into account this variability of the process. And it's only when you fully control your process that you can begin to narrow your specifications. But once again, it has to be linked to the activity of the protein.
Sure. I mean, how do the regulators view that in terms of assuring patient safety if you're building in that variability?
Yeah. The regulators will ask you to have specifications that are based on your clinical data and on your manufacturing process variability. So clearly, at the beginning of your development, you can have quite broad specifications that you will narrow if possible at the end. But yes, once again, the specification have to be linked with your data, your clinical data, and the activity and the manufacturing process.
Okay, thank you. Gordon, can I bring you in there? Would you like to add anything to this aspect of manufacturing control strategy and the quality of the product?
So this is something which we also opened at the beginning. We have to be careful not to kill the entire glycoengineering process by over-regulating it, because at the moment, we don't know which parts are essential and which are not. So we need much more research. We need to generate much more data and then look back at the data and see what is actually essential and what is not.
Okay, super. I think I'd like to leave that topic there. I think that's been well answered, and just move on to the last few questions. We're coming close to the end, and I'd like to cover a slightly different aspect of this Glycan landscape that we've been discussing, and address this question first to you, Gordon. Are glycopeptides the most informative molecules to analyze for glycodisease markers?
I think this will be very disease specific. For example, if we are talking about things related to inflammation, yes, we would have to look at the IgG, either glycopeptide or glycans. But for example, we have a very good biomarker which predicts diabetes in incident diabetes, so the future diabetes. And here, released glycans from the total plasma proteome are still more informative than any of the glycoproteins and glycopeptides we looked at the moment. So some of the diseases will be protein specific, and then we have to look at the glycopeptide level. But other diseases, metabolic diseases like diabetes, will not be so protein specific. So if we average across all proteins and look at free glycans, we will actually get more prognostic potential. So I would say here depends on the disease.
Okay. Would you like to add anything to that, Mike, at all with your expertise?
No, I mean, Gordon has done an awful lot of work in that area, and I think he's answered it, very well.
Okay, all right. Well, I'm going to stay with you then for the next question, if I may. This question is: how can glycome information be better integrated with other omics data to give insights into disease pathways and more complex multi-omics markers?
Yeah, that's a very interesting question. I mean, the thing is that if you consider the omics like genomics and proteomics and so on, they are template-based, right? So from a genomic data, we can get amino acid data and amino acid sequences. We can't do the same thing with glycans because it's not template based. It's based upon the type of glycan that you get from a cell will depend upon the enzyme activities which result from those proteins being produced. There is a lot of interesting work, I think, in terms of trying to predict the type of glycoforms that you get and the heterogeneity glycoforms you get from reaction kinetics, reaction networks, looking at things like key enzyme activities and also substrate availability for cells. And I'm talking about bioprocesses now, of course. And that work is developing very nicely, I'll say, to the point where eventually I think there'll be some degree of predictability in terms of, okay, you add certain substrates, you know what certain activities are, we can predict what glycoforms will come out. And that will help an awful lot. Things like the manufacturing perspective of, forming, different biologics that you might want to produce, particularly biosimilars that I'm thinking of. I think that space has been actively researched and will develop, I think, nicely in the next few years.
Okay. Can I come back to you, Gordon, with your expertise in this general area? Would you like to make a comment on that?
Yeah, so this is still a huge challenge. So we had two large European networks, each of them with 15 plus partners, and we worked for 5 years, each of them. And we still do not have a good method how to integrate the multiple omics data. So we have tens of thousands of people with the different omic data, including the glycomic. And we have published a number of fancy papers showing that you can integrate this data, but I still haven't seen a good bioinformatic approach in integrating glycomic and other omic data. And one of the key problems is that there is a huge scale difference between the glycomic data, where we generate 100, 150 variables, and then you have 20 million single nucleotide polymorphisms or thousands of different mRNA transcripts. So we intensively work on that, but we still do not have a solution. I think one of the key problems is that the majority of people doing other omics are still ignoring glycomics. So the large databases like UK Biobank or All of Us Research Project, they don't have any glycomic data. And once we will have glycomic data in these big databases, and when they will be put in a public domain, then there will be more people trying to solve this problem, because this is still an open challenge, how to integrate different omic data.
Okay, thank you. Maybe we can expand on that on the final question I have for the panel. This is the final question. And I'm going to start with James, if I may, and I'm going to go around everybody. What do you believe are the future hot topics in glycan analysis? And are we limited by technical capability or basic research in understanding and controlling glycosylation? So I'm going to start with you, James.
Again, I'll touch on going back to Glycan. I think that would be it. If I know there's a few PhDs in place working on the potential for having a robust released method to mimic that of the N-glycan, and have, again, touching on, as everyone has mentioned so far, the link between glycan structure and the impact of glycans on the therapeutic antibody or product, and having more information around that, I think, would be my main interest, at least.
Okay, thanks. So that's your dream, James.
Yeah.
So, okay, super. I'm going to come to you next, Gordon, if I may. I'm sure you have some interesting things to say on this topic.
So I think the key problem and a challenge we still have is that we have no ideas how glycosylation is being regulated or even executed. So we did a series of so-called genome-wide association studies only for IgG, and there are over 40 genes which regulate IgG glycosylation. So the enzymes which actually do the work are only minor component of this entire glycosylation machinery. So, and also we have no ideas, for example, how FAB and FC glycans can be differently regulated. Of course, there is availability of different sites, but how can they be differently regulated based on hormonal changes and so on? So I think there are a lot of basic research has to be done. And then the second thing is our analytics is still not sensitive enough. We are just scratching the surface of the glycoprotein. We are analyzing few glycoproteins. We are still not close to the single cell glycomics and not even single glycoproteomics. So I think we are, when we look where genomics was in the 90s, this is where we are today. So I think we need this big push, which happened also in genomics. We generated all these next generation genomics technologies to have the next generation glycan analysis technologies, which will enable us to really understand the glycobiology and to see how can this help us understand the disease.
Right, thank you. That sounds like we still got a long journey ahead of us. Mike, if I can come to you next.
Yeah, well, I mean, in terms of the future, I would like to focus on, the biosimilar market, which I touched on before. I mean, in terms of therapeutics, a lot of good monoclonal antibodies are out there, but it does now, many of them coming to the end of their patent time. So, there are going to be more and more companies, producing biosimilars. And the big challenge for them is to look at the originator molecule, not have the cell line, which the originator company had, and try and be able to reproduce not only an amino acid sequence, but a glycoform pattern, which is equivalent. So how is that done? Well, it's done through control of the bioprocess. It may be done through glycoengineering and so on. But I think the tools towards being able to mimic a preferred glycoform, if you like, I think that's an important topic which I think will be tackled in the future.
Okay, thank you. And finally, I want to give Arno the last word on this. Arno started us off today, and I'd like him to give his views.
Yeah, so I agree with what has been said so far, but we saw that we have many methods available today. There are some pitfalls in some methods. We know that glycans are not easy to analyze. Site-specific analysis is something that is quite challenging too. But anyway, we have a good toolbox. But the main issue we have, and Gordon said that earlier, if you send the same samples to many labs that use the same method, you won't have the same output at the end, and you may have some differences in terms of results, and this should not happen. So I think we still have to work on the robustness of the workflow in itself, not only the analytical part, but also the identification, the bioinformatics, and so on. And the second point, the analysis is one thing, but we need also to better understand what is the role of the glycans on the mode of action of the biotherapeutics. And once you know that, you will have a better understanding on what you need to do in terms of analysis, what is the level of information you need. In some cases, the glycosylation analysis could be quite quick. In other cases, you will have to go deeper into the glycan analysis to understand better what happens and what is the impact on the potency of your future drug.
Okay, thank you very much, Arno, for your last comments there. This brings our discussion on glycans to a close, and I'd like to say thank you to all of you who have joined our workshop to hear our experts. And of course, I would like to thank our fabulous expert panel wholeheartedly for sharing their expertise with us. Have a great day, and thank you.
Well, thank you very much, Mike. What an excellent discussion. I'm sure all of you watching and listening have enjoyed the conversation, and hopefully you've learned something from our experts today. Talking of which, a big thank you to Arno, Gordon, Michael, James and Mike for taking part today and openly sharing their thoughts, insights and experiences into the issues surrounding glycan analysis in biopharma. Don't forget, if you still have a question that you haven't submitted, you can still do so by clicking the Ask a Question button below, and we'll forward them all to Arno, Gordon, Michael and James, who I'm sure would be happy to follow up with you afterwards. Thank you also to our scientific partners in this event, being Agilent Technologies, for all their technical input and support. This discussion is due in significant part to their involvement, and I would encourage you all to visit the Agilent Technologies website to find out more about the instrumentation offerings and solutions in this field. Finally, I'd like to thank you all for attending this event today. I hope our panels have widened your knowledge of the various fields covered and helped you move your Glycan analysis testing forward. Thank you very much, and goodbye.